And I'm Pasharamuna Khabar.
Today we are going to talk about a comparison of bio-fermented solution from fish waste, shrimp waste, and crab waste, on water quality and growth rate of sea grapes.
So about the introduction, sea grapes are superfood that incredibly rich in mineral and amino acids, while being low in calorie and fat. However, growing is a huge challenge because it's so sensitive to water quality like pH, DO, salinity, and temperature. This makes production very expensive, which unfortunately keeps local, small-scale farmers out of the market. At the same time, we are facing a massive environmental issue with seafood waste from fish, shrimp, and crabs that end up polluting our water and damages the ecosystem. Our goal is to bridge these two problems, finding a way to turn that waste into something valuable.
And our solution is to transform those seafood byproducts into bio-fermented liquid to act as a natural nutrient boost for the sea waste. In this study, we test how different types of organic fermented liquid affect both the growth of the sea grape and the quality of the water. We found that by using these recycled nutrients, we can significantly improve the weight and length of the seaweed while keeping the environment stable. Essentially, this research isn't just about growing the better seaweed. It's about reducing pollution, adding value to waste, and creating a sustainable, low-cost farming framework that local communities can actually afford to use in the future.
So there is the question, does water quality change when sea grapes are colored using bio-fermented solution, and if so, how? Do bio-fermented solution affect the growth rate of sea grapes, and if so, how?
So the hypotheses are water quality obtained from cultured sea grapes using bio-fermented solutions shows change, and bio-fermented solutions affect the growth rate of sea grapes.
Material and equipment. We are going to use pH meter, salinity meter, dissolved oxygen meter, thermometer, plastic basin, plastic baskets, oxygen generator, digital scale molasses, microbial activator, fermentation tank, and light.
The study site looked at learning center from sampling of sea grapes and seawater was conducted within the designated study site and sent the data into the globe data entry analysis of growth performance. The water quality parameter analyzed in this study include pH, DO, salinity, and temperature statistical data.
So, making biological fermentation. First, combine 2 liters of clean water, 1 liter of molasses, and 1 kilogram each of fish, shrimp, and crab scrape in the container. Then stir the mixture until it becomes a homogeneous mixture. Next, add the microbial activator to mixture at a ratio of 1 gram per 2 liters of fermented liquid. Next, ferment for 15 days. During fermentation, open the lid and stir mixture periodically. And after 15 days, filter out the food scrap from the fermentation liquid.
Prepare the experimental set. Acclimate 1 month old sea grape for 3 days, then cultivate it in 20 aerated basins. For treatment, 3 replicates each were applied. Control and pre-bioferment shrimp, crab, and fish at 20 to 35 degrees Celsius.
Step for raising sea grapes. Place it in a location with direct sunlight for 6 hours and replenish with seawater every 7 days. In experimental set 2, 3, and 4, fermented liquid was added to seawater at a ratio of 1 milliliter per 1 liter of seawater.
Data collection and recording. Water quality test. First, measure the pH value. Next, measure the water temperature. Then, measure the DO. And finally, measure the salinity. Measure the width and length of the sea grapes and record the results.
And the results, water quality study. Figure 1 show a bar chart of average pH value. Figure 2 show a bar chart showing average DO value. Figure 3 show a bar chart of average salinity level. Figure 4 show a bar chart of average temperature. Figure 5 show a bar chart displaying average weight of sea grapes. And Figure 6 show a bar chart displaying the average length of sea grapes.
So, conclusion. Water quality improvement. pH remains stable and neutral across all groups. DO highness in treatment 3. This is due to craft calcium carbonates acting as a pH buffer and a slower decomposition rate, which minimizes microbial oxygen consumption. Salinity highness in treatment 2. Likely from the release of accumulated marine minerals and salts during shrimp-based fermentation. Temperature consistent across treatment, averaging 29. Group performance treatment 3 outperformed all groups in weight and font length. Group ranking: the first being crab, the second being fish, the third being control, and the last being shrimp. Chitin serves as a vital nitrogen source and natural growth stimulant. Mineral, calcium carbonate, and essential minerals stabilize pH and reinforce the seaweed's cellular structure, promoting superior development.
And this is impact to community.
And this is acknowledgement.
And the reference.
Thank you.